Hello, everyone, and welcome to this new webinar. This is the fourth in a series together with I want to say it right, with the International Safe Cassava Network. We have today as a chair for this session, Ross Gleadow, who is the coordinator for said network. I will leave you with her, and I will be back together with Kira Maher as well during the Q&A to assist. So Ross, the floor is yours.
Great. Well, thank you very much, Isabel, for through the Global Plant Council for hosting this webinar. I'm Ros Gleadow. I'm Professor of Plant Biology at Monash University in Melbourne in Australia. And I'm the, I guess, the chair of the International Safe Cassava Network. This network is a new incarnation, if you like, as some of you may know, of this cyanide and cassava diseases network that was started by Howard Bradbury and lapsed when Howard passed away. And it really brings together people interested in cyanide and cassava and safe consumption. So it's an interdisciplinary group. It's cross-cutting or intersectional, if you like. And the aims of the group are to capture the knowledge that we already have about konzo and cassava, and that includes, we've been interviewing some of the leaders in the field, the founders of the field, people like Julie Cliff, who I can see is online today, who have really done a lot of pioneering work in the field. It's also about discovering new research that's going on, and that brings us to our webinar today. And the aim is really to look at ways to develop safe cassava. So we have everybody involved in this network from many, many different countries who are either, dietitians, plant biologists, and medical people, and so on. And we are grateful to the Global Plant Council for hosting us. Just before I introduce our guest, I just want to acknowledge, as we do in Australia, the traditional owners of the land on which I'm meeting, the Wurundjeri people of Waiwarong language group, who are part of the Kulin Nation, and acknowledge their people and their elders, past, present, and emerging, and pay my respects to them, and also pay respects to any other First Peoples on whose land you may be meeting wherever you are in the world. With that, I'd like to just now introduce our speaker. We can go into a lot of details. Matt Bramble, I met coincidentally through this International Safe Cassava working group, which we had established as an ad hoc group to steer this network. And he's a health scientist. He works in neurology and looks at the molecular systems that underpin konzo. He spends a lot of his time in the Democratic Republic of Congo, which is not an easy place to work in. And interestingly, last year actually saw an outbreak of konzo as it was happening in real time. And really interestingly, he's won many awards, and he's recently been selected as a Pew Biomedical Scholar to continue this work in lower middle income countries. And this is a huge honour and is likely something to bring konzo to the attention of the wider world, which I think is really important. I won't say any more, Matt, because I find your research so interesting, but I'd just like to pass over to you, and I'll just jump in if you need anything.
Awesome. Thanks, Ross, for the intro. So I will start sharing my screen now, Isabella, yeah? Full screen, everything okay? Okay, well, I'll get started. As Ross said, I'm Matt Bramble. I am based in Washington, D.C. When I'm in the U.S. at George Washington University and Children's National Medical Center. Today, we're going to talk about a few things I've been working on over the last 4 or 5 years, which is kind of emerging to show that oxidative stress and the gut microbiota are likely contributors in cassava-associated neuropathies, and in this case, konzo, which is what I'm most familiar with. I know a lot of the people that come to these seminars have heard a lot of this before, but I see a lot of people that I don't recognize. So we'll do a little background before we get into some of the new science that I've been working on. If you're not familiar, konzo is a non-progressive neurologic disorder. Based on the phenotype, it's thought to affect upper motor neurons. We see this condition predominantly in higher frequencies in children and women of childbearing age, which is a loose term. Childbearing age could be anywhere from, I suppose, 13 onward. It's most prevalent and really restricted at the moment to sub-Saharan Africa. Of course, I say here that thousands of cases have been documented, but it could be predicted hundreds of thousands of cases certainly exist. Failed to detect speak. Okay, I apologize. I have no idea. Okay, so as I said earlier, it was most associated in sub-Saharan Africa. DR Congo and Mozambique are the two countries with the highest burden of disease. It's associated with the consumption of bitter cassava, coupled with general malnutrition, and it's been speculated that there's also a deficiency in sulfur-containing amino acids, such as cysteine and methionine. This disease comes in outbreaks, which is unusual, but it's just like a normal outbreak, and you see these in times of drought, famine, and war. Anytime that agriculture and livelihood is disrupted, konzo seems to pop up. There are multiple impairments with konzo, but the most visual and striking is the lower extremity phenotype, and it's variable in different stages. So you see these two individuals on the right here with the disability. This is very classic of konzo. In addition to the physical disability that you get just from diet, there are other issues such as vision, eye issues, and speech impairments, things that have been recently studied that are associated with konzo. And some have actually found that there are severe cognitive impairments as with individuals with severe disease. While konzo itself may not directly kill you, it certainly results in extreme morbidity and eventual mortality. Obviously, being paralyzed in rural villages are certainly no good recipe. If you haven't seen konzo, what I'm going to show you now is a video, and this is it covers the 3 stages of konzo, and these again are not progressive. So you either get stage 1, stage 2, or stage 3, and this is what it looks like. So this is stage 1, which is considered mild, but as you can see, it's certainly not very mild. And stage 2 here is a bit more dramatic, and it's really only classified currently as, do you need a walking support or do you not need walking support, as stage 1 and 2. And stage 3 is the most severe, and unfortunately these children have to crawl around villages. So you can see just from the phenotype that the symptoms are quite extreme, and to think that all of this kind of stems from their diet. And the diet that is likely to associate with this disease is cassava, which is the staple for well over 500 million people. It's native to South America, and it came, I believe, in the 1500s or perhaps earlier with the Portuguese to Africa, and it obviously thrives there. Typically, they grow 2 main varieties. It's considered sweet cassava versus a bitter cassava. And really, this is just a delineation of toxins in the plant. So bitter cassava, which is associated with konzo, has high levels of cyanogenic compounds, or compounds that can eventually turn into cyanide. Now, it's pretty well established how you remove these cyanogenic glucosides from cassava. And if you're not familiar, essentially, you start with your cassava plant on the left. You harvest the roots, which are tubers, kind of like potatoes, but this is the cassava network, so I'm sure you know what cassava is. You peel the husk and you soak it either in the river or, as you see here, there are these soaking pits. And these are part of the problem, because as you soak the toxins out of cassava, you eventually intoxicate or saturate the pit, and therefore the toxins don't leach out as well. And then you dry the product, and you either pound it into flour and turn it into fufu, or you can ferment it into different products. Ideally, the process is to remove these cyanogenic glucosides before you get to the final product and ingest it. But of course, different processing methods and depending on water availability, you can certainly have high levels of toxin remain in the final food product. So within cassava, the cyanogenic glucoside the primary is called linamarin. And linamarin is just a glucose attached to a cyanide moiety. And essentially what happens is when you ingest linamarin in the gastrointestinal tract, it can actually be converted directly into hydrogen cyanide. Obviously, a potent poison. But if you have sufficient sulfur amino acids, such as methionine, particularly cysteine, you can detoxify this, this compound into a less toxic form known as thiocyanate. And this is via an enzyme known as rhodinase. So the detoxification of cyanide is pretty well conserved across all species, obviously due to its deadly nature. But it does require correct balance in order for the body to be able to detoxify cyanide into thiocyanate. And this essentially is the detoxification hypothesis of konzo, that children have an inability to detoxify. And we will discuss this in more details. A lot of the groundwork has been laid in konzo as far as the epidemiology, the neurology, the associations from, some on this talk, such as Julie Cliff, Desiree Chala, Brad Berry, and others. But in the last, 8 or so years, I've wanted to kind of move into the more biology omics aspect of the disease, because that had not really been looked at into great detail. So for this talk, we're going to go over two interesting findings using metagenomics, where we look at the gut bacteria composition of individuals with konzo. And we're also going to do overview of proteomics studies that I look at, where I was trying to find biomarkers that associate with disease, both of which turned out to be pretty interesting. So originally, our first question was, could the gut microbiome be implicated in the disease? It makes sense. Everyone in the areas, in these villages, consume a cassava-heavy diet, but only a fraction of people are affected with konzo. So it's not exactly 100% penetrant, so to speak. I say here it occurs in some, 10% of populations during outbreaks, but these numbers vary. We see villages where the percentage is much higher and others where it is much lower. But because the microbiome is highly variable between individuals, we thought it was a good investigation. And we also know that the gut bacteria play a large role in processing xenobiotics, medicines, toxins, you name it. But this idea, of course, was not mine. This idea that cyanide could be modulated by the gut has been around, I say since the 80s, but I believe even earlier in veterinary studies, since cassava is also a staple crop for livestock. And, a classic example of how the gut microbiome actually influences a dietary toxin is the desert wood rat. This is always my go-to and my favorite. So this rat lives in the California deserts, and it eats a toxic plant known as creosote. Creosote is like the waterproofing stuff you'll see on docks, and it's kind of like a tar. But moral of the story, it's very toxic. And researchers were very unsure of how this rat eats a monotonous diet of creosote but doesn't die. So they did a microbiome study, of course, and they found, interestingly, that there was an enrichment of bacteria that were able to metabolize aromatic compounds. And if you see here on the left, creosote is certainly an aromatic compound. So it kind of led to the notion that this gut bacteria in the rat was actually responsible for breaking down the toxin. So if you think of it in the same concept of cassava, you eat cassava, it has a toxin, maybe the gut has some type of role in this. It makes sense. So what we did is we took a large cohort of individuals from Kinshasa in DR Congo, which is the capital. Down here in Kahemba is essentially the epicenter of konzo in that country. And then we also sampled from a rural village known as Masimanimba, which very similar diet, but they don't seem to get konzo. There are other terms in here, high prevalence, low prevalence zone, but generally speaking, let's just think of, we have an urban center, a rural area without konzo, and a very rural area with high prevalence of konzo. And we did a large microbiome study on these individuals. So we looked at the bacteria in the stool of, several hundred children. And when you first look at it, I say the bird's eye view here, each circle represents a group of Kinshasa, Masimanimba, Kahemba. And essentially, you have to rely on statistics for this, but statistically speaking, it's easy to distinguish, who came from Kinshasa, who came from Kahemba, who came from Masimanimba. So there are three unique bacterial profiles. So that, this is a classic statistic approach, Bray-Curtis type analysis, which is old school ecology studies. But of course, we wanted to see if a computer could actually distinguish between the gut bacteria profiles of individuals from these three areas with accuracy. And what we see here on the left, you see accurate, area under the curve, or in this case, we can think of it as accuracy, over 90%, pretty much for all. So the classifier was very accurate in determining if I give it a sample, it could tell me with high accuracy that this person is from Kinshasa, this person is from Masimanimba, and this person is from Kahemba. So that was pretty interesting in the sense that now we kind of establish that there are indeed unique microbiome profiles between multiple areas in Congo with and without konzo. So of course, the real question in that study was, do differences exist in gut bacteria structure between children with konzo and children without konzo in Kahemba? However, to our surprise, there were actually no differences between these individuals. So in this panel here on the left, you can see certainly non-significant for either. And this was from a low prevalent zone of konzo and a high prevalent zone. But the moral of the story here is that children with konzo and without konzo from the same area almost look identical. And kind of shocking to me at the time, but that's okay. And you can see here in figure D on the left, the computer random forest classifier also had a very poor accuracy. So if you think chance, that's 50%. So this is 60. So 10% above chance was able to distinguish. So that kind of told us that there likely is not a big difference between the microbiome within the region of Kahemba. So children with konzo, children without konzo, virtually identical. So this, as I go through the literature, because I was certainly no expert in gut microbiome at the time, I'm still not, but I find a lot of studies where they find lactic acid bacteria, which are always our good bacteria, particularly Lactobacillus plantarum. They've shown that these bacteria are very effective at taking linamarin and releasing cyanide. It can break that bond. That bond requires a beta-D-glucosidase enzyme. Not very common in the mammalian digestive system, but very common in bacterial species, particularly the fermenters. So I got interested to see if some of these known bacteria with cyanogenic potential were actually enriched or depleted in these microbiome studies. So the four panels here on the right in this figure are Kahemba. Just think of that. The last four are always Kahemba. So in this study, I looked at particularly, it's called Lactobacillus or Leuconostat menasterides, Lactococcus lactis, and Lactobacillus plantarum. And these were the three strongest bacteria that could release cyanide. Coincidentally, they're also the three main bacteria that ferment cassava. So you can kind of see the connection. If you're fermenting cassava, it's filled with these lactic acid bacteria, you're consuming them. It's possible that this interaction is releasing a lot of hydrogen cyanide from the linamarin. And we actually find here that in Kahemba, where konzo is most prevalent, these lactic acid bacteria are significantly upregulated. Since we did metagenomics, we actually had the power to look at individual genes specifically. And what we find here in this last four panel again is that the gene required to take linamarin and convert it into hydrogen cyanide is actually increased in children of Kahemba. So all of this is kind of going together, showing that perhaps that the children in Kahemba have bacteria that actually exacerbate the effect of consuming linamarin. And when we look at a village where I say they have the same diet, but no cassava, Masimanimba, we actually see here in green, they have an increased abundance of bacteria with the enzyme rhodinase. And as I said earlier, rhodinase is the enzyme that you need to detoxify cyanide. So it looks, in this study at least, that there are regions in Congo, Kahemba, that may have bacteria that are detrimental, and others have bacteria that are positive, or detoxifying. So essentially, this leads to potentially a susceptibility scenario where you consume fufu with high amount of linamarin. You happen to have gut bacteria that's enriched for beta-glucosidase positive bacteria, such as the lactic acid bacteria I just showed you. As a result of having that, you're going to increase your hydrogen cyanide production. You have a limited amount of detoxifying bacteria, and effectively, you're going to have a scenario where you are not detoxifying enough cyanide into thiocyanate, so therefore you are building the toxicity component. Alternatively, you could have the protective scenario where you have bacteria that are high in rhodinase, the detoxifying enzyme, which would subsequently reduce your levels of hydrogen cyanide and increase your levels of the detoxified product, thiocyanate. Now, of course, is this 100% going to be the case every time? No, but the idea that the gut microbiome could modulate konzo was pretty interesting at the time and still is. And our work kind of developed this detrimental protective role of the gut microbiome in relation to konzo. Now, the gut microbiome doesn't give you konzo because obviously we all have a gut microbiome, but it's very possible that it could modulate konzo in some way or another. So over the years, we've used multiple approaches to try to better understand the biology of konzo. We've looked at the gut microbiome, we've looked at DNA methylation, proteomics, some genetics perspectives, but the biological mechanism of how you ingest linamarin and you end up with a motor neuron disease remains unknown. So given the unknown nature of the biology of the disease, we wanted to investigate if we could identify biomarkers in children who are affected. It's been searched for a while, but a reliable biomarker had not really been established, to the best of my knowledge. Since Julie Cliff's on the phone, she'll know best. So what we did here is we used in 2011, Desiree Chawla here at the bottom, who is my mentor in the konzo space, had collected samples from a cohort of individuals with konzo and individuals without konzo. Here they're unrelated. And we prepared these samples, the plasma, 11 years later. It's always, a little risky to do such a study, I think, but we did it anyway. Took these plasma, ran them through a particular mass spec, did some fancy analysis, and what we found is that there were indeed proteins that were different between affected and unaffected, 28 in fact, so not a lot, but a good start. We see here that the computer algorithm in panel C is able to distinguish the groups pretty well, and these are based on markers of inflammation. So this is also very interesting in the konzo space. But what we had found here, most importantly, if you look at figure B, there is a protein off here to the side, which is downregulated, the most downregulated protein in konzo. And this protein is called GPX3, or glutathione peroxidase 3. At the time, I had never heard of glutathione peroxidase, let alone glutathione peroxidase 3. So with some digging, we, I found what it was, and I'll explain shortly. But for the first study, nothing really stuck out to me. Nothing made sense, really. There's some proteins up, some proteins down. What does that mean? So I wanted to see if some of these findings were reproducible. So in 2021, I collected a sibling cohort. So these are children affected with konzo, and then their corresponding sibling from the same household who do not have konzo. We did a much larger study here, 222. This time we changed the mass spectrometer and did fancy analysis. And this time we only found one protein that was different between siblings. Again, that protein was GPX3. So now I had to begin believing that there was something to the story here. And given our last cohort, the size was big enough, so we were able to look at stage 1 konzo, stage 2 konzo, stage 3 konzo. Regardless of the stage, GPX3 was the only significant difference between affected and unaffected. So now, I was getting pretty excited that we had found a biomarker of sorts. So looking into glutathione peroxidase and the whole glutathione peroxidase system, it's actually very interesting in the space of konzo because the glutathione peroxidase system is our body's innate antioxidant defense. What glutathione peroxidases do, and there are 8, and the vast majority are intercellular. So during normal cellular process, you're always generating reactive oxygens and peroxides. So these enzymes are scavenging those molecules very quickly before any damage is done. But in the plasma, there's only one GPX3, and that's one GPX, and that's GPX3 that circulates in our plasma, extracellular. And what GPX3 does, and the others, is they use glutathione to reduce reactive oxygen species and peroxides so that damage from oxidative stress does not occur. What's interesting about this class of proteins is that they are selenoproteins. And a selenoprotein is one that requires dietary selenium to be made. And dietary selenium is a micronutrient that we often get through meats and some nuts, particularly Brazil nuts. However, meat and Brazil nuts are certainly lacking in the diets of individuals that are affected by konzo. And we also know that cassava harbors nearly zero selenium. So it's very possible that you can see a scenario here where the diet itself may affect your antioxidant system by not having adequate selenium. Again, I didn't know any of this until a few years ago. Never heard of GPX3, selenoproteins, but now I'm becoming an expert, so don't worry. So after we had found that GPX3 was affected, we asked, well, are other proteins, if you target mass spec, it has much more power once you know what you're looking for. And we also find in that last cohort of the siblings that seleno-P was also significantly downregulated. And seleno-P and GPX3 are the primary selenium reservoirs in the plasma. So if you ever go to the doctor and you're diagnosed with a selenium deficiency, unlikely, but if you are, it's probably because these two proteins were low. And this is kind of how you determine selenium deficiency. Well, that's interesting. I have a hard time still understanding how one sibling in the house is selenium deficient and the other four are not. That seems it doesn't really add up because the household diets are very similar. The mama prepares the food for the family all at once. So it was a hard story to tell. But moral is that there was certainly a downregulation of selenium proteins in children with konzo, which are very, very important for oxidative damage protection. We did a few additional type of experiments to validate this. So these are different, an ELISA-based approach to look at GPX3. So we don't need a fancy mass spec. We can just do an ELISA. And it's a little small here, but essentially, if you look at stage 1, stage 2, and stage 3, these dotted lines on the distribution are the 95% confidence intervals. So essentially, the 95% confidence interval for GPX concentration in someone with konzo versus without aren't even overlapping. So they are distinct entities, which led us to a recent publication where we highlight that glutathione peroxidase 3 is a potential biomarker of konzo. Now that we've looked at it in 2 different cohorts, 10, 11 years apart, it was very obvious that something was there. And I'm not the first to have thought the antioxidant role in konzo was important. Desiree and others have looked at, they've shown that isoprostane markers, which are signs of oxidative damage, are also elevated in children with konzo. It's pretty well known that cyanide can induce apoptosis via oxidative stress in different models, from neuron cells, actual chickens, and the list goes on. So it's well established that cyanide certainly causes a reactive oxygen cascade. So when cyanide binds to the mitochondria, it inhibits respiration, which generates a lot of reactive oxygen species. And therefore, you certainly need your glutathione peroxidase system to be working very effectively in order to prevent damage from this oxidative stress. So to remind you, we talked about the detoxification hypothesis earlier, where I said that sulfur amino acids are important for detoxifying cyanide. You need a sulfur donor to turn cyanide into thiocyanate. So this is one aspect. The other hypothesis that's kind of revolved around konzo development is what I call, I guess, the glutathione hypothesis here. Glutathione is the reducing agent that glutathione peroxidase uses to remove the reactive oxygen species. However, cassava certainly does not provide enough adequate sulfur amino acids. We know that. They've actually shown that cyanide metabolites can actually reduce glutathione, already setting the stage for reduced antioxidant capacity. And interestingly, cyanide is a tripartite system here, and cysteine, which is the sulfur amino acid that you need to detoxify, you also need adequate cysteine to generate glutathione. And the original paper that kind of got into this sulfur concept was done with Julie Cliff and colleagues, where they had looked at sulfur content, I believe, in urine. Don't worry, Julie, I promise I read the paper a while ago. And kind of extrapolated that this was going on. But I don't know if anyone had looked at direct plasma levels of amino acids until this study. And when we look at children who are affected in blue and those, or children who are affected in orange versus the unaffected sibling, most importantly, we'll come down here to cysteine. Neither affected or unaffected children had any adequate levels of cysteine in this particular study, which was very unusual. So based on the cysteine being reduced, we can, one, assume glutathione production is not at its highest, and two, detoxification potential is also not at its highest. So you can kind of see where pieces of this antioxidant puzzle are coming together, which could make children susceptible to konzo. And this is kind of leading to this emergent theory that I have and others have on konzo. So we'll kind of go through this here, and I'll show you just a few more things, and then we'll end. So essentially, in this theory, you're going to ingest linamarin through your diet. In the digestive system, the bacteria containing beta-glucosidases are going to lyse linamarin and release hydrogen cyanide. When this happens, kidneys and liver will detoxify cyanide into thiocyanate under the assumption that you have proper sulfur amino acids, which we know these children do not. When this detoxification is inadequate, the cyanide itself is certainly going to attack all mitochondria that exist in our system, either in our vesicle system, our organs, our brain. Anywhere cyanide comes in contact with, it's going to cause a reactive oxygen storm. So now we're inadequately detoxifying cyanide. Now we have cyanide attacking mitochondria, releasing all kinds of reactive oxygen species. And there are systems in place for when this happens. In this case, GPX3 in the plasma. So as reactive oxygen species are getting out of control, GPX is, gobbling all of this up in a normal scenario. However, as I'm showing you that these children have, sometimes it's 70% reduced GPX3, this system here is inadequate. So now in the plasma, now we're having an oxidative storm that's kind of leaking out into the plasma, going all over the body, and certainly can damage, in this case, motor neurons, which are the biggest and most vulnerable to oxidative damage in the first place. But also what happens in this system is that it can also get out of control. So when the plasma becomes filled too much with reactive oxygen species due to a GPX deficiency, certain enzymes can actually convert cyanide directly to cyanate, which is a well-established neurotoxin because it requires an oxidative environment. Hemoglobin itself can actually take thiocyanate and convert that back into cyanide, which could further propel the situation. So essentially, what I think is that as your antioxidant system is challenged, in this case, a reduction in GPX, perhaps glutathione, you open the system up for essentially a storm that can no longer be contained until you suffer severe neuronal damage, perhaps what's happening in konzo. Now, all of the things I showed you were tested in individuals that had had konzo for some time. So I didn't know if if GPX3 being downregulated and cysteine being reduced was a product of having the disease, or if this is perhaps a reason why you got the disease. So as Ross said earlier, last year I was in Congo and notified that there was likely an ongoing konzo outbreak happening just about 8 hours outside of the capital, very close. It's usually never so close to the main city. So me, some friends, and Congolese team, we head out to the village and we investigate. We take samples. We do some educational initiatives with different ministries of health and nutrition, where they teach the mothers the wedding method and where they kind of explain what's happening and why individuals are getting konzo. So here we were able to, in about 6 days, we collected 250 or so individuals. They came for the study. And I've never seen konzo early. These children within, some within 5 days, some within a month, but everybody was within a month of contracting or developing the condition, which was very sad because in places like Kahemba, they're very familiar with konzo and they know what to expect. Whereas in this village, Bongo Yasa, they had never seen konzo before, so no one was sure that, what was going to happen. Unfortunately, these children are going to be left with lifelong disability, and they're going to end up like the video I showed you, all simply from eating the cassava. But in this new study of early onset, this is really where I can ask questions. Are there differences in antioxidant capacity when you get konzo? Is there different nutrition when you get konzo? Is GPX3 an actual biomarker? A whole bunch of things that can be asked in a new cohort that I could never ask in someone that had konzo for 5 years due to too many variables. Now this has happened last year, so things are going a little slow, but I do have I put it together yesterday, so it's not pretty figures, but some interesting things. So the question was, is GPX3 a biomarker for recent konzo onset? And you can look at this volcano plot here, and this is a flip. So if it's on the right, it's down in konzo, and if it's on the left, it's up in konzo. And what you find again in children that just got konzo, GPX3 is the most significantly downregulated protein again. So this is the third time we find GPX3 as the most likely biomarker of disease. And seleno-P, which is the selenium transport protein, just like our previous 3 studies, identical. However, what we find here in this new study, which is very exciting, is that there is a protein over here to the left, which is very upregulated in children with konzo, and this is called MT1G, or metallothionein 1G. Never heard of it. When you go down the rabbit hole, you actually find that this protein has been a well-established marker of neurotoxicity, which is very interesting in the scientific space. So the fact that this protein is upregulated in children with konzo may indicate, for the first time really, that there is indeed neuronal death, and due to the apoptosis of these neurons, it's actually leaching out this MT1G protective factor into the plasma, because otherwise it's not really seen much in the plasma. In fact, in controls, it's almost 0. And in children with konzo, it's, well, certainly not 0. So this is the new proteome of this acute study I looked at last year. And one, it confirms all of the things we found before, and also opens the possibility of very interesting biomarkers that may really shed light on what's happening in konzo, because it's very well established that there are neuronal, motor neurons have died, but it's MRIs are, we haven't really seen the site of death of cells, and we certainly haven't really detected markers of cell death. But this MT1G may be that. We also were able to look at glutathione. This was the prediction. Remember, I said based on cysteine levels being low, I would expect glutathione levels to be low. However, what we find here is that, so the normal range for glutathione is between 1 and 10 micromolar or millimole. I think it's micromolar. For today, it's micromolar. So between essentially 1,000 and 10,000 here on the scale. And we find that both affected and unaffected, so orange and blue, while low levels of glutathione, they're actually still within the normal range on average, as you see, which I was very surprised about. Glutathione works in conjunction with its reduced form or oxidized form, GSSG, and this measures oxidative stress. So the normal range is 1 to 100. So 1 glutathione to 100 GSSG is the ratio. So that's a normal range, 1 to 100. So despite them having low normal levels of glutathione, they actually have very low levels, very low ratios, which would indicate that both control and konzo are in some type of oxidative stress situation based on just this one marker. We also looked at cysteine again. We did the same amino acid profiling of this acute case, and we find here, unlike the last study where both affected and unaffected were low. We see only children with konzo have suboptimal levels of cysteine. You can see the normal range is 9 to 47. The kids here are averaging, 6. So kids with konzo really have no cysteine as predicted. When we look at the other sulfur amino acids, methionine, the same case is true. Normal range 13 to 45 micromolar. The kids are ranging here maybe at 10, the average and below. So both sulfur amino acids in this new outbreak where the children have only had konzo for a few weeks, very low. Glutathione and the ratio, not what I expected, but also very interesting and unusual. There's a lot more studies to be done. Like you see, this is my to-do list. So we're actually working through all of this. I'm a little slow sometimes, but we're getting there. But I wanted to show you some, I haven't shown this to anyone yet. So some new updates that really, I think, begin to confirm that oxidative stress, oxidative damage, GPX3 is certainly part of the biological mechanism of konzo. And on my end, I always think, science is fun, and understanding the biology and science of konzo itself in a box is very interesting. But unfortunately, these are, real kids, and the way to solve konzo is not in my lab or anyone's lab. It's really trying to end the severe poverty and poor agricultural practices. That's the only way to end up in konzo. This little guy here is named Caleb, and you can see Caleb over here on the left. You can look at Caleb's knees and understand the kind of life that konzo gives you. The kids are very happy, and in a village such as Kahamba, when 20 or more % of kids that you see are severely disabled, it becomes normal. It's not weird that I have all the kids hanging out with me, eating on the ground, and it's completely normal for them, and it's really sad when you see it. So the idea of the science that I like to do is heavy science to understand the biology of the disease. But really, it's to have talks like this and to actually expand people's interest in konzo, because many haven't heard of it yet. Scientifically, it's a very interesting disease, and it covers gene and environment. It covers neurology, metabolism, microbiome, pretty much you name it. Konzo can be part of it. So I think through scientific awareness and finding interesting things, people become more interested in the disease. And ultimately, I think, as we garner interest, we eventually solve konzo the clear way, of how to do it, as you see here. So with that, I think I'll end and then take some questions and thank my collaborators and, of course, the foundational people. All of this stuff, all of these works have been built on Desiree Chala, Julie Cliff, Thorkild, and others, of course. It's not a huge field, but nevertheless. So if you have any questions, let me know. And I guess, Isabella, you want to take over, or?
Yeah, me or Ross.
Yeah.
So
Yeah,
thank you so much, Matthew.
Of course.
Yeah. That was absolutely amazing, and has certainly really shown me, given the whole. Sorry, I'm just trying to get my screen up again differently. Incredible. Just gobsmacking. The 20% of kids in this village are disabled. That's an incredible high proportion.
Yeah, don't quote me on that percentage, but a lot.
But it's more than 1 in 100.
It's something you see when you go into the village. It feels like half the town is unfortunately having to crawl around. It's not that much, but I've never seen so many disabled people who aren't thought of as disabled, because everybody's disabled. If everybody is the same, then no one's really disabled. So it's a very odd situation.
Yeah. Well, that was really just most interesting for me as a plant biologist, completely out of left field, but I'm making connections.
Yeah, it's all easy. It all goes together.
Yeah. All this sort of oxidative stress stuff, brilliant. And also, I think also verifying, if you like, work that Hans Rosling and Julie Cliff and others did on that paper you showed, where clearly the sulfur that's required for making thiocyanate was important for the diet. But this connection with selenium to me is just like completely out of left field.
Yeah, selenium seems very, it would be nice if selenium deficiency is the cause of konzo, because something like that is intervenable in theory. If this is a genetic cause, where there's some type of genetic variant that reduces GPX expression, which has been found particularly in ALS, another motor neuron disease. That's a little harder, but that could identify children at risk. Whereas if selenium is the problem, you could, imagine a scenario where you enrich for selenium in the diet. It still requires an international effort, and it's just as hard, I think, to provide selenium as it is to provide new food. Somebody has to bring the compound, somebody has to, it's really not effective. There has to be agricultural practice where you're growing selenium-rich foods, or eating, chicken eggs, something like that. Like I said, Brazil nuts are number one, but they're not growing Brazil nuts. I thought you could plant Brazil nuts and Brazil nut trees in Congo. I'm like, oh, it's a rainforest, except they take like 100 years to grow. So not going to work for this one.
Yeah, and a lot of allergies to Brazil nuts as well.
Yeah, probably,
and also selenium is toxic if you have too much of it, so you can't.
I found out, too many Brazil nuts, you get selenium toxicity. I'm learning a lot about the plants, Ross.
Yeah. Okay. So what I will do is I know there's some questions in the Q&A. So Isabel, do you want to read through those, or do you think I should take those? I think you could do that.
I can help you.
Yep.
Yes. So there is a question by Darner Dufour. It's very short and to the point. Why isn't konzo a progressive disease?
Hey, Darner, I'd have to say, scientifically, that is the weirdest aspect of the whole scenario to me. Because after you get konzo, essentially you get, unfortunately, even poorer. If you have a family of 8, you have 4 kids who are have a severe disability, well, you can imagine the burden on the family that is already struggling. So your diet gets worse. You probably eat more cassava, cyanide-rich cassava. So I biologically have no idea how in the world the disease doesn't get worse. In theory, once you get konzo, it should only go downhill from there. And I see one of the questions that say, why doesn't it progress, right? Oh, no, that was Darner, never mind. Yeah, so why it doesn't progress, I have no idea. Thorkild notes that what could happen is you could have a second attack. So you start with stage 1, and there's another drought, you're intoxicating yourself tremendously, and now you have another event, and now you have stage 2 or stage 3. But I think of it kind of like a stroke, Once you have one stroke and, half your side's paralyzed, that doesn't really get worse. It's final. That's where it's fixed. You could have another stroke. So the stroke concept is kind of how I think of konzo here. Something happened, almost had some kind of maybe an antioxidant induced stroke, killed some neurons. But maybe you have to reach a threshold. To reach that threshold is difficult, perhaps. So imagine, next week it starts to rain, you start eating some nuts, your selenium goes up, your GPX goes up, even though you're still eating bad cassava, maybe your other factors are a little higher, so you never cross the line again. That's the only thing I can think of, but it doesn't make any sense to me how it doesn't progress and how it doesn't, progress. Yeah.
Go, Ross.
A quick comment from me, just to follow on from that, and from Dinah's comment. So if you say kids who get like stage 1 konzo, I mean, Julie will probably be able to answer this if she's online. She can jump in or put it in the chat, Julie. Can they get it again? Like, can you get then another year, another harvest, or another drought when the cyanide's high? Can they get worse again? Can they go from stage 1 to stage 3? Not because the disease is progressing, but because you've got it again.
Yeah, I think there have been reports of that happening, a second attack kind of thing. But it's not, like you're saying, it's not the disease is not progressing. The environmental factors or whatever has caught, wherever you're at has given you an opportunity to further worsen that area. And also why it's just that particular motor pathway. Because when you examine the kids, I had a colleague, a pediatric neurologist, who came out on this last trip, who had never seen anyone with konzo, and he was certain that he was going to find something that's been missed, some new feature, except it's not. All the reports on konzo is exactly as it is. There are no real peripheral things. There's no sensation problems. It's just this gate and this pathway. So why those motor neurons only is strange. Also, there's a lot of strange aspects to the disease.
Okay. Sorry, Isabel, you're going to move on?
Yes, we have more questions. We have one question from Mary Fitzpatrick. Did you consider testing the cassava leaves in an area in Eastern Democratic of Congo? The flora was very low in cyanogens, but the leaves very high and more associated with thiocyanate in the urine than the flower. So did you consider testing the cassava leaves?
Yeah, I did not test them, but I did test the flower, which was not all, actually. It was, I should have shown that data. It wasn't like everybody's cassava was poisonous. Again, this was a month later. Who knows what happened in that moment? But the leaves, for sure. I think the leaves are indeed the harbor of linamarin, but the way the leaves are prepared traditionally, it's pretty much boiled down to the foundation. So during this process, you're really getting rid of the linamarin, and the hydrogen cyanide is coming off. Anecdotally, Desiree Chala used to tell me that they used to test how poisonous the cassava was by taking the leaves given to the goats, and if the goats died, then you knew. I don't know if this is true, but it sounds kind of interesting. Then if the goats died, then you have a very toxic crop, because it is very poisonous. But I did not test. No, is the answer. But I presume also high, considering the flower was higher than one would want.
Yeah, I think that the next question really comes on from that. Jim just thanks you for the presentation. Yeah, it's great. But do you know what the levels of the cyanogens in the cassava were? I mean, you said the flour. Did you test that? I missed what you said.
Yeah, so I ordered some of the kits from Jam in Australia to test the flour, and I can't recall the exact numbers, but they were not. They were all not off the chart. The village one of the villages did take us down to where they soaked the cassava, and visually it looks like your classic problem. They have the cassava soaking in this cauldron, this pit, which is, I did take a pH of that pit. It was 2. And as the pH drops, cyanide also cannot be liberated. So the processing system here, so if you acidify your soaking pit, you are not detoxifying your cassava. So even if you put your cassava in for a week, it comes out nice and soft, ready to dry, ready to grate, except it didn't take into account that pit was pH 2. So nothing is really being liberated out. It's still a toxic product. So yeah. So the measures of the root itself, are always pretty high from Congo. And it's tough to change processing. If you just, like in Massima Nimba, I said they don't get konzo, but this area where I'm studying, in the microbiome study, Massima Nimba was my control area where they ate the same diet but did not get konzo. This new outbreak, Bongayasa, maybe 20 kilometers from Massima Nimba. So what I said in 2021 is no longer true. This area is indeed konzo prone, just like the region is Bandundu. So it seems like that whole region is certainly susceptible. It's a plateau. It's kind of dry, but in Massima Nimba they have a river, and they tend to soak the cassava in flowing water. So this pH acidification doesn't happen. That could also be a reason why konzo is low. Whereas Kahamba, a little higher up on a plateau, a little more arid, water's far, they soak in the pits, which get acidified quickly. But in Bongayasa, they were soaking in pits too, not in a river, which I guess has become, I see a pit of cassava pH 2, you can almost guess somebody's going to get konzo soon.
Yeah, actually, I can't remember who it was, someone in the cassava workshop who'd been doing some work in Congo, was it Joel? But they were saying actually because at some of those high elevations, the temperature's not so high, cyanide's volatile at 26 degrees. So
exactly.
if it's only 22 degrees, things like your wetting method aren't going to work very well.
Yeah, it's the devil's in the details. Yeah, just like that. If your pH in your soaking water is too low, you're not removing compounds. If the temperature's too low, the sun's out, but you're not reaching where you need the volatility. So it's, that's why I say konzo, the science is interesting. The agriculture, there's so many components to the disease. It's probably very simple, a simple recipe, but how you get to that recipe, it's like, all roads lead to Rome, but everyone's a little bit different, and each one can get you to the same disease.
Jim just had a follow-up question saying he was actually referring to the level of the cyanogens in the root, but it sounds like you didn't measure that. Also, from a number of people have worked, including ours, what's in the leaves is not a good indicator of what's in the roots either.
Mm-hmm. Yeah, I didn't this time, Jim, but we have indeed measured the root. Actually, in Philippines, Vivian Topor, I think she's actually on she's the one that showed me how she did it in Philippines, and as expected, the cassava roots in konzo or in Congo raw are very high in cyanogens. But again, they're growing the crop with that feature. They want that. It's a great pest deterrent. So a lot of people always suggest putting cassava in without linamarin. And I believe they tried this, and what happens is essentially, if you're growing a plant in the heart of Africa without any pest deterrent, well, the bugs are going to eat that plant down to the root. So then you starve to death. So you have to grow a plant that is very toxic, one that bugs don't like, two that thieves also don't like, because you have no idea what kind of poison, you're dealing with. So this is kind of how this is developed, that the plant's natural pest defense is, resulting in neural damage, but also providing the chemical that you need to feed everybody in the area, because you need this poison to keep pests, predators, so on and so forth away. So it's a real catch-22 here with, cassava is great. It feeds, a lot of the world, except, ... kind of thing.
Yeah, which is the point of safe cassava. It's an absolutely brilliant crop, but we just have to make sure it's safe for people. Lucas asked the question, does this need to be taken into consideration for animals, because the cassava peel is used to feed animals? I think you showed a paper on chickens in one of your examples.
Yeah, that's where a lot of the microbiome work kind of came from. So it's pretty well known, if you just took cassava roots, like the actual tuber, and fed to I think it was pigs in that one study, you could kill pigs, and pig is a big animal, with the root. So farmers have known that you don't feed animals raw cassava, essentially because it will kill. But all the microbiome foundation has kind of been built on cattle and ruminants. So they know that there are, if you feed cows certain things, they get bloat and they swell up. But that's a reaction of the gut bacteria with some chemical. They knew the same thing for cassava. They were finding high levels of thiocyanate, which could only be done really through bacteria. Then if you take the contents of the, one of the cow's ruminants, take the liquid out, mix it with linamarin, you start making cyanide. So that was very strong evidence that there are bacteria in the gut that can take that sugar and lyse it into cyanide, which you could kind of see developing in a, and it's decently known that if you kind of take this lactic acid bacteria and mix with the cassava, it kind of speeds up the detoxification process, because the lactic acid bacteria is effective at lysing linamarin. However, in the microbiome study, you can think you eat it all day, and that's all you eat. You're actually eating a lot of this lactic acid bacteria too, whether it's a resident of the gut or it's something that you're just consuming daily. As you consume it, and it's inside of you as well, and if you have any residual linamarin, now all of that good bacteria that you've eaten now can eat the linamarin as well, letting hydrogen cyanide be released. So, all the probiotics are lactic acid bacteria, but, of course, this could be one of those, what do you call it? Canary in the coal mine thing. It's great until it isn't. And this is a case where perhaps no lactic acid bacteria would be favorable.
Right, good. Well, that's interesting. Now, Pedro asks, do you know if there are any initiatives to generate cassava varieties with low levels of cyanogens using either transgenesis or gene editing?
Yeah, I believe it's been done. I'm pretty sure there's linamarin-free crop. Jess, I don't think she's on the call, but she would know. But I'm pretty sure it's true. And talking with Vivian Topor in the Philippines, she certainly works with low linamarin crops, and they have a particular strain where it produces what's that in here? Beta carotene. So the root's a little colored, so it actually gets some beta carotene in there and has low levels of cyanide. So I think crop replacement is a good idea. All of this has to be tested. I've always wanted to take some of the good cassava that's grown down in the Philippines and plant in Congo. And but I always think of a scenario where I plant it in a different soil and it turns to the most poisonous cassava that ever existed, and I end up causing more problem. Agriculture is very and linamarin and cassava is very sensitive to the environment. So when it's a drought, cassava is making the linamarin is more concentrated if it's very wet. So I could take wonderful cassava from Philippines, take that and stick it into some arid plateau in Congo, and it turns right into bitter cassava and equally poisonous. So it's not an easy crop to, low levels of linamarin in Philippines may not necessarily mean low levels of linamarin in the plateau of Cahamba. It could be as poisonous as the stuff they already grow, which is probably why it is so poisonous. The soil is unfavorable. It's dry. There's a lot going on.
So that's good. Thank you for that. Is konzo or konzo-like disease in indigenous communities in South America, like in South America, where they eat bitter cassava?
I think I had a call with Darna Darfur, and I think I asked this, and I think, quote me if I'm wrong, Darna, that I don't think you see anything like this down in South America. And I find that also fascinating. I think there is a study to be done there, but it's also wet where the cassava is grown. It's certainly a rainforest down there. There's plenty of water. Processing is a bit different. I believe Darna has talked about before. So I think that's a classic different environment. Same crop, different environment. You get this. You don't see konzo up in the rainforest of Konzo or rainforest of Congo much. They of course eat cassava as well, but there's 1,000 rivers and the Congo River and a different environment.
Yeah, different environment for sure. Now, Artur asks, thank you for your excellent presentation and the work. He joined the webinar late and he missed the information about the period during which your research was conducted. So I think there were two periods, is that correct? So during what period was your research done? I think there were two periods, did you say? The
microbiome was, or the proteomics, the GPX, there were samples from 2011, 2021, and then last year, 2014. So multiple cohorts, multiple age different, years, conclusion always the same. Microbiome was done in 2018-19, and there will be a follow-up study to that published kind of soon, where I look at the siblings as well. I like to do the experiments multiple times. I have a hard time believing my own work, so I like to do it over and over to make sure it's right. Like the GPX, I can say it's a biomarker, but if I don't show you 5 times, are you going to believe me? So good quality data is important in really shaping how you mitigate any disease before, you say don't eat cassava. Well, you can't say that. Maybe it's not the cassava. Maybe there's an inherent issue with the individuals that get konzo, because 90% of people eat cassava. They have high levels of urinary thiocyanate. They get intoxicated but they don't get konzo. It's not like the children who did not get konzo have low levels of toxin in their urine. They're off the chart too, which Julie's found, Desiree's found. Everyone's looked at. Unaffected children have just as high levels. But so there must be a biological factor that tips the thing. Maybe there's GPX, maybe selenium, but something like that. There's more to it than just the plant.
So I think there is a comment here from Jim coming back again on that other thing and saying in Amazonia where he works, no one detoxifies bitter via via fermentation, like the process you describes. Yeah, I think that must be very different from using the TPG process. He's saying what contribution to konzo does this. Yeah, so what what contribution to konzo does this detoxification process make? I think you've probably covered that.
Yeah, I think in theory it helps, right? If you have a pit, you fill it up with bacteria, it eats up all the linamarin. In a controlled environment, I say that's great, of course. But when you're hungry, and you have 5 kids, you can't really. One, you're not really testing to know if you really got rid of all the toxins, which you really never do. And 2, if I have 5 kids that are starving and my cassava has only been down 2 days, well, I got to pull that out and start making a flour because I'm out of time, kind of thing.
Yeah.
Yes. Yeah. I mean, konzo really is just a disease of poverty. Being poor is the risk factor for konzo. That all other things are, if I go to 200-pound me goes to, I could eat all the bitter cassava probably until my eyeballs fall out, bitter fufu. I'm not getting konzo. You fatten up everybody, they're probably not going to get konzo either.
Yeah, I think there was a famous paper that Julie wrote, I think in 1992, I think, on the konzo being the war and famine crop. And
exactly.
I mean, cassava being that and konzo being a consequence of disease. I think given the time, because we're an hour and 15 minutes and we know that the people have other things to get on with their day and those of us in Australia need to go to bed, I think this has been a really interesting discussion. It has been recorded, and in a few days' time, it will be. Isabel can give us the details. Will be posted on the Global Plant Council YouTube channel.
Okay.
Matt, I really just want to thank you for this, because I think this is just shows what this network's about. Is that we need to bring people from very, very out there disciplines. To me, what you do is sort of out there compared to what I do, like growing cassava and measuring cyanide.
I had to learn, all of that stuff from you. So you're the other aspect. You're the front end aspect of the disease.
Yeah. So the plants are my thing. So thank you very much. And what I'd like to do is to just pass back to Isabel to close the session.
Okay, and you can feel free to share my email, Isabel. If anyone has questions, I'm sure I think someone said they wanted to contact me, but yeah.
Yes, I will send everyone in a couple of days. Everyone who has registered to these webinars, I will send the link to the recording. I will include as well your email address in case they are interested in contacting you, and I will also include extra information on the webinar in September and our symposium in October. So you have something to look forward to. And
perfect.
Thank you everyone for attending this webinar, staying until the very last question, and see you in a month's time. Ross, do you want to add something?
No, just thank you everybody. Stay tuned for the symposium that's coming up. We've got sessions on food systems, cassava the plant, things on konzo, Torquil's organising that, and also on public health. So Umberto from Mozambique who's organising that. So keep an eye out for that. It'd be great to have people involved. So thank you, everybody.
Thank you.
Bye-bye.
Bye, guys.